Good afternoon, everybody. Welcome to Asphalian's webinar, Signal Management Process in Veterinary Pharmacovigilance, Key Points. Before starting, a couple of things to take into consideration. The webinar will last for 30 minutes. In case you have any questions, please submit them in the chat and we will reply them by email at a later stage. We will also send you the presentation per email as well as the link to the recording shortly. And now it's my pleasure to introduce our speaker today from the Pharmacovigilance Unit, Laura Eder. Laura is Drug Safety Associate Director. She has a PhD in biomedical research, as well as a background in biology and biochemistry, and a master's degree in health administration. She holds solid experience in pharmacovigilance audits, medical writing, and in overall pharmacovigilance activities. In today's webinar, she will cover essential aspects, including the critical stages of signal identification, prioritization, evaluation, and communication, providing the foundational knowledge for an effective signal management system. Our colleague Alicia will talk briefly about Asphalian's safety services.
Thank you so much, Lydia. Welcome everybody to our webinar, Signal Management Process in Veterinary Pharmacovigilance, that, as you all know, our expert in veterinary vigilance activities, Laura Eder, will be hosting. Before turning over to Laura, if you all allow me, I would like to take a few minutes to briefly introduce our work here in Asphalion. We are a leading regulatory, scientific, and safety consulting company dedicated to assisting pharma and biotech companies. We support our clients through various stages of product development, from initial phases to achieving marketing authorization. We take great pride in our team of experts who are genuinely committed to each and every project. We deeply value and strive excellence in everything we do. Regarding pharmacovigilance activities, Asphalion acts as a one-stop shop, providing expert support through whole product life cycle integrated in global context. Asphalion started providing pharmacovigilance services back in 2007, and since then, we have been growing and working to stay up to date with the latest safety regulations. Currently, we are providing support to more than 80 clients in more than 160 active collaborations. We are proud to say that our pharmacovigilance system is outsourced for more than 20 companies. We act as European KPPV for more than 15 companies. We perform literature searches for more than 215 APIs, and all these activities are performed with the highest level of quality. Regarding veterinary vigilance activities, we can provide global support, ensuring that your system complies with the European regulation. We are responsible for maintaining and performing vigilance activities for more than 60 veterinary products, markets in Europe, Latin, and Asia, including managing adverse events, performing literature searches, or preparing signal management reports, among others. Let me also highlight that we count with veterinary experts, not only in our vigilance department, but also in other units, including lifecycle management or development writing, allowing us to provide global and transversal support. And now, I will hand over to my colleague Laura, who will surely help us understand how to develop a successful signal management system. Go ahead, Laura, please.
Thank you, Alicia. So welcome everybody to this webinar in which we will review the key points of the signal management process in veterinary pharmacovigilance. Let's start with the agenda. Today, first of all, we want to talk a little bit about some veterinary regulatory news, and then we will focus on signal management. First of all, we will do a brief introduction about what is a signal. Then I will give you an overview of signal management process. We will talk a little bit about the complexity of the signal management process, about the frequency of signal detection, and then we will move to the signal detection procedure. We will finalize the webinar talking a little bit about signal validation and dose factor, signal assessment, and signal notification. So now I pass the word to my colleague Paula, who will explain the new regulatory updates.
Hello, thank you very much, Laura, for introducing me, and good afternoon. My name is Paula, and as part of the Asphalian's Lifecycle Management Unit, I would like to give you a brief overview of the latest regulatory news for veterinary medicinal products. First of all, I would like to comment on the new update of the QRD template. This is the version 9.1. So marketing authorization holders must submit the specific variation requiring assessment so that it can be implemented by the end of January 2027. This is applicable for all types of procedures. Then we continue. In this case, applicable only in Spain with the new SIGRE authorization, which includes the management of veterinary medicinal products for domestic use. This is new because until now SIGRE only managed waste from human medicines, so it will be necessary to join SIGRE as a scrub by the end of the current year. Another update would be the additional use of the IRIS platform when managing variations requiring assessment. And this will be effective from January 2025 for EMA procedures. And then finally, I would like to mention that the web-based electronic application form will be issued on Q1 2025 for testing. The final date of implementation is still to be defined, but we really encourage you to follow the latest updates on it and start preparing internally for the changes. I hope this clarified the more relevant upcoming updates. And well, if you happen to have any questions, do not hesitate to contact us. Thank you very much and enjoy the webinar.
Thank you, Paula. So let's start. So what is a signal? Well, as you can see in this slide, a signal is defined as information that arises from one or multiple sources, including observation and experiments, and which suggests a potentially new causal association or a new aspect of a known causal association between an intervention and an adverse event, or a set of related adverse events that is justly likely to justify further investigation of possible causality. New aspects of a new known association may include changes in the frequency, distribution, duration, severity, or outcome of an adverse event. A signal, it is often related to all veterinary medicinal products containing the same active substance, including combination of products. But it is worth mentioning that certain signals may only be relevant for a particular product or in a specific indication, strength, pharmaceutical form, or route of administration, while some signals may apply to a whole class of veterinary medicinal products. So in practice, for signal involving spontaneous reports, usually several case reports are needed to trigger a signal, and only in very rare cases would a single well-documented report concerning a serious adverse event or involving several animals be enough to trigger a signal. In addition, it's important to have in mind that a signal is a hypothesis. In the process of signal detection, we look for information in order to confirm or refute that hypothesis. And in case we confirm the hypothesis, we have to perform an evaluation in order to determine if the signal detected is a safety concern, and also we have to decide the actions to take. So I have structured this figure for the veterinary GBPs because I think that it perfectly reflects the main step of a signal management process. So as you can see, the signal management process consists of, but not limited to, the pharmacovigilance activities of signal detection, prioritization, validation, assessment, and recommendation for actions. The outcome of the signal management process could be a change to the benefit-risk balance of the veterinary medicinal product concerns on animal health and welfare, or public health, or on protection of the environment. Here we have some situation. The marketing authorization holder identify an emerging safety issue. In that case, the MAD has to notify it to the relevant competent authority without delay and no later than 3 working days following their notification. Other situation is that the MAD identify a change to the benefit-risk balance or a new risk. In that case, the MAD shall notify it without delay and no later than 30 calendar days to competent authorities. And this, as you can see here, can lead to an update of the product information or to establish any other risk minimization measure. Another situation is that the available information is insufficient to conclude a potential causal association at present, but further information is expected to provide evidence that could change this conclusion. In that case, it is expected to perform a close monitoring or a post-marketing surveillance study. We'll go through this in upcoming slides. So, everybody knows that the signal management process is very complex, as there is no standard way to organize the process, and also it will depend on each organization, large, small companies, type of products, health authorities, and so on. Although there are guidelines for conducted signal detection, there are no rules, and one must be flexible. Besides, clinical criteria should always be applied, meaning that prior knowledge about the product, the animal treated, and the disease to be treated is needed. So in this slide, I want to highlight the key elements of the signal that fits this complexity. So first, we have the frequency and the signal calendar that we have to elaborate. As you know, it's very important to create that calendar based on the recommended EMA dates and thus efficiently plan the preparation of the signal reports. Another point is to gather source data collection, specifically high-quality information. Other point to consider are the methodology to use that we will see later on, the clinical judgments to apply. And on the other hand, once a signal is detected, a complex step is the identification of actions to take. And of course, the notification in timely manner. And finally, we cannot forget all the quality requirements to be followed. Let's focus on the frequency of signal detection. As you know, all marketing authorization holders shall conduct at least one signal detection analysis per year for each of their products included in the UPD. As mentioned before, grouping will be allowed on the basis of same or similar products. In order to facilitate and coordinate the evaluation of the competent authorities, due date for submission of the annual statement on the benefit-risk balance are set up for all active systems concerns. These are defined annually, although more frequent submission could be specified by the INC. The signal detection analysis shall be performed within 2 months before the annual due date. It's important also to take into account that in case of emerging safety issues or upon request from competent authorities, signal detection can be performed ad hoc. And IRIS will be available to submit the signal detection 15 days before the due date. So, in practice, on an annual basis, the marketing authorization holder shall monitor at the end of each year the EMA publication of the recommended due dates and prepare the corresponding annual signal detection calendars. So let's move to the signal detection process and let's focus on the source of information to be analyzed in a signal detection report. Here I have listed the different sources of information that we have to take into account. These are the data warehouse, the PB database of the holder, literature, health agencies, the medically important terms, because as you know, these terms must be prioritized. Also, we have to take into account validated signals. This means new validated signals within the period, previous validated signals or closed signals within the period, and also the sales data. And how we are going to analyze all this data. So it is recommended a combination of quantitative and qualitative methods. In case of quantitative methods, these are the statistical analysis, and the qualitative methods refers to the traditional methods. So focus on statistical analysis. This analysis, the sources of this type of analysis are the data warehouse and the PB database. This type of analysis have good benefits, some benefits, like that they allow to analyze a big number of data simultaneously. They are repeatable and consistent. They are customizable. They are auditable, and also they help us to identify a potential signal. But there are also some risks associated to this analysis. Why? Because they generate many false positives. there could be a high impact of duplicates, and they do not imply a causal relationship, and they do not establish a clinical significance. So, for this reason, traditional methods are complementary to these statistical analysis. And the traditional methods allow us to confirm or discard association of signals previously detected, and also allow us to perform a detailed evaluation case by case. Which are the sources of the traditional methods? The sources of information are line listings, individual cases, associations of adverse events per Petra category, trend analysis, literature searches, regulatory intelligence, or social media. So now I want to focus on the key analysis in signal detection. Here we can talk about three. The first one is the proportional reporting analysis. This is a quantitative analysis based on ROR, and it is mandatory once a year in data warehouse. Also, a key analysis in signal detection is the individual case analysis and the case series analysis. In this case, this is a qualitative analysis based on the review of data from individual and cumulative cases in the database. And it is necessary each time a potential signal is detected. We also, another key analysis, we can find the reporting rate analysis. This is the analysis of reported cases versus exposed animals based on sales data, and this is necessary to contextualize the signal analysis data. So let's focus on ROR, the proportionality analysis. Well, ROR indicates how many times greater the probability of an adverse event being reported within our medication compared to the probability of it occurring with other medications in the database. It's important to keep in mind that this is a statistical association, as I said before, and does not imply a causal relationship. Therefore, a case review and clinical judgment must always prevail. Here we have two hypotheses. Hypothesis one is that the combination product event is notified with higher frequency for our product than other products. In this case, is ROR major than 1. The null hypothesis, that is the combination product event, is not notified with higher frequency for the product than for other products. Another, in that case, ROR is minor or equal to 1. And then we have the confidence intervals. For the hypothesis to be statistically significant, the lower bound of the confidence interval, lower bound equal to ROR negative, must not contain the value 1. And it cannot be equal to the null hypothesis. So, as you can see here, the ROR formula is a 2-per-2 contingency table, in which A is the number of reports for a specific adverse event associated with a specific veterinary product, B is the number of reports for other adverse events associated with the same veterinary products, C is the number of reports for the specific adverse event associated with other veterinary products, and D is the number of reports for other adverse events associated with other veterinary products. I have also included in this slide the threshold ROR analysis that we have to include in the data warehouse. So, I just introduced data warehouse. Data warehouse is used for the signal management analysis, which is mandatory to be reviewed for all veterinary products. And here I have listed the most used queries. And now we are going to review them in depth. Let's start with the adverse event overview. Well, the adverse event overview will be the first one to be analyzed. A quantitative analysis will be performed in order to detect a potential increase of incidence of cases during the review period compared to the previous one, as well as for fatal cases. As a general rule, 3 case reports are needed for signals concerning medically important Petra terms, or 5 cases report for signals involving other Petra terms. However, as I mentioned before, flexibility should always be applied, and this should not be understood as a strict rule. In addition, analysis will be focused on the species for which the concerned veterinary product is indicated. And the quantitative analysis is always performed at PT Vitra level. Also, Sometimes it's needed to perform the analysis within a group of PT that belong to different SOCs, but they are related to, for instance, a similar thing or symptoms. An example of this could be the bovine neonatal pancytopenia. Finally, the selected PTs after this analysis will be selected for further analysis. So let's focus on the raw analysis that I introduced you before. So now, as I just mentioned also, the PTs previously selected will be evaluating following the raw analysis. Again, as a general rule, PTs with ROR negative major than 1 or ROR major than 2 during the reporting period, and 3 cases involving MEI terms or 5 cases involving other viral trends are selected for further evaluation. However, very important, there could be exceptions, such as situations in which only 2 cases are reported, but there is a high number of animals affected. So in those situations, those cases must also take into account for further analysis. Also, you must have in mind that other PTs that meet ROR criteria should be also selected. Finally, it's also important to consider the PTs related to a potential signal and review the cases to see if the reports include only one PT or multiple PT. And why? This is because this helps avoid duplicates in the evaluation while ensuring that we gather cases related to the potential signal. So let's move to line listing. If the raw criteria is met, the corresponding line listing shall be obtained. And the evaluation of those listings shall be focused on this, on the quality and nature of the information, proportion of animal treated versus animal affected, number of animal died, previous medical history, relationship between exposure and response, causality assessment, listlessness of the events reported, seriousness of the events, and also other exclusion criteria can be applied. For instance, cases could be discarded due to events being clearly related to a different dose or route of administration. Or one other criteria for exclusion could be either a duplicate case or the event being clearly related to other suspect drug or as a consequence of concomitant diseases. So let's move to the traditional methods. As I said before, the statistical methods that we have just seen are complementary to the traditional analysis methods. And these ones, the traditional ones, are used to confirm or refute association detected through the previous methods, the statistical ones. Meaning that the traditional methods are always necessary. So these traditional methods include case analysis, literature searches, and other relevant sources. Let's start here the case analysis. Well, the criteria that an event must meet to be a potential signal are mainly based on detecting new event, the MEI terms, and other analysis. So let's start with the detecting new events. Let's focus on the term of reporting frequency. Well, events with an increase on the reporting in the period or first time reported will be candidates to be a potential signal, as it means that new information has been received for such event. Again, as I mentioned before, as a general rule, 3 case reports is needed for signals concerning MEI terms, 5 case reports for signal involving any other federal term, but as always, flexibility should always be applied. Then we have another concept, which is listedness. Listedness refers to the classification of the adverse event based on whether it is already documented in the reference safety information. So terms that are listed in the reference safety information are already known to be associated with our product. Therefore, the events mentioned in the section 4.6 of the reference safety information shall be considered listed. Another point to analyze is the expectedness. Why? Because if the event is not literally listed in section 4.6 of the reference safety information, but it is related to an event already listed or equivalent, we must apply a clinical judgment to decide if the report event could be classified as unlisted but expected. Then MI terms are events that are medically significant or known as to be associated to the veterinary product, and these events will be always prioritized during the signal detection process. Other analyses will be to perform a frequency analysis when there are 3 or more cases reported during the interval from the same PT, regardless the listedness, and these events in that case will be considered as potential signal. Similarly, changes on the outcome, unexpected lack of efficacies, and any other aspect related to the product may be taken into account to consider a certain event as a potential signal after making the medical judgment. And very important is to review cases with related beta trends. As I mentioned before, literature search, we have to review also literature searches and other sources of information like social media, qualitative facts, safety information, or safety alerts from different health authorities, and previous validated signals. So, we have performed the signal detection process. What is next? Next is the signal validation. Signal validation means to verify that all the initial available information related to the detected potential signal contains sufficient evidence demonstrating the existence of a new potential causal association or a new aspect of a known association, and therefore justify further analysis. Once it's detected a potential signal, the first step in the validation process will be to perform a first quality check of the cases and the evidence supporting a signal based on our previous experience. So, as a minimum, it is expected to check at this step that the event occurred after exposure to the medicinal product, that the signal is not based on duplicate, that the suspect adverse event is not already adequately reflected in the current product information. Other information that should be checked at this step is if the signal concerns an increase in the number of reports involving an expected event, and that this increase is not related to an increase in sales volumes or other external factors. We have to talk a little bit about those factors when we are talking about the validation process. Those factors is used to know different packages of a product, which allows to know how many animals were treated. The dose factors determines the average number of animals of a particular target species, which can be treated by one package of a given packed site, regardless of the formulation. The appropriate dose factors shall be provided for all volumes of sales data and target species per package identifier and per country. Those factors shall be calculated for potential signals as part of the calculation of the adverse event incidence and signal validation process. The dose factor shall be a positive numerical value with or without decimals. And also the methodology used to calculate the dose factor, this is very important and you don't have to forget it, shall be recorded in the Annex 4 of the PSMA. It is strongly recommended that changes to dose factor shall be strictly limited over time. Those factors shall only be changed if significant changes are made to dosage regimen. For instance, changes to antimicrobial dose rates. Here I show you the common formula to be used in signal detection. But nevertheless, it's important for you to know that other can be used depending on the product analyzed. So, once the analysis of the previous awareness, the validation step is completed, and no conclusions are pulled out, a second route for collecting additional information will be initiated with the aim to evaluate if the valid signal is considered as a new risk or not. The assessment of the signal should be as comprehensive as possible in order to reach a high-quality decision and signal outcome. The assessment should include a cumulative review of all available evidence, and in this cumulative review, The available pharmacological, preclinical, clinical, and epidemiological data from all sources shall be reviewed as applicable in order to conclude on a potential causal association between the veterinary medicinal product and the concerned suspect adverse event. So here, I want to show you some important elements, some key elements to consider. First of all, we have the total number of cases. We have also the incidence. We have also to consider additional cases reported with related terms. For instance, other pediatric terms indicating clinical complications or different states of the same reaction. Other elements to consider in the signal assessment steps is the consistency of the evidence across cases, quality of the data and the documentation, dose reaction relationship, possible mechanisms based on biological and pharmacological possibility, disproportionality of reporting, potential drug-drug interactions, also additional cases from products of the same class of medicinal products, information of epidemiology of adverse reaction or the underlying disease, databases with larger data sets, and also information from other regulatory authorities worldwide. So, once we have performed this signal assessment, as a result of the assessment of the signals, what is next? Well, we should conclude whether the available evidence review supports a potential causal association or not. If it is concluded that the safety profile of the product has changed, the need for additional risk minimization measures and any other regulatory activities shall be considered, including a variation of the terms of the marketing authorization. So this leads to the following possible actions to be considered. First, the available evidence support a causal association resulting in a change to the benefit risk or a new risk. Here we have three situations. We can have an emergency safety issue, a validated signal with the need of regulatory actions or risk minimization measures, or a validated signals without further regulatory action. On the other hand, we can be in a situation in which the available information could suggest that a potential causal association is at present unlikely. In that case, we have a signal refuge, and we don't expect further actions beside the routine pharmacovigilance activities. And then we also can have the situation in which the available information is insufficient to conclude a potential causal association at present, but further information is expected. So in that case, we have to perform a close monitoring of the situation and maybe also a post-marketing surveillance study. So let's finish the webinar with the signal notification. So after the procedure, the final steps is the signal notification. So here we have two separate procedures that can be identified and that will require evaluation by the competent authorities. First of all, we have the veterinary signal assessment report following the EMA template to report the evaluation of validated signals that require further regulatory action. This report is submitted continuously throughout the year by the marketing authorization holder, and this includes emerging safety issues and signals where a new risk or a change to the benefit-risk balance is identified. Then we have the annual statements confirming that the signal management process has been conducted and confirming that the benefit-risk balance of the concerned product is unchanged. This procedure can also include the annual submission of the validated signals assessment through the year, which did not require urgent attention or did not lead to any proposal for further regulatory actions. But in case no signal was detected or validated, a standard statement confirming that the signal management process has been conducted in line with the GPPs and also confirming that the benefit-risk balance of the concerned veterinary product is unchanged, it's sufficient. To finalize, as mentioned before, in order to facilitate and coordinate the evaluation by the competent authorities, due date for submission of the annual statements on the benefit risk is set up for all active substance concerns. The submission is done through the EDIS portal, and don't forget that the EDIS portal is available 15 days before the due date. So this is the end of this webinar. I hope you enjoy it, and do not hesitate to contact us in case you have any doubt or you need our help. Thank you very much for attending this webinar.
Thank you, Laura, for this excellent presentation, and thank you to all the attendees. We hope you enjoyed the webinar, and remember, you can send your questions anytime at info@asphalion.com. Have a nice afternoon, everyone. Bye-bye.
Bye.